Thank you, Leo. Thank you for inviting me, and thank you all for being here early in the morning. It's a real pleasure to present you my work. Let me just get familiar with the buttons. Yes, okay. I already went too far. Okay. In my talk, I will show you evidence that TMS-evoked potentials can be used to study the physiological activity emerging from specific structural cortico-cortical connections. Sorry. Okay. Before that, I just want to briefly introduce what TMS-evoked potentials are for those of you who might not be familiar with them. First, TEPs are obtained from TMS-EG co-registration, as Leo said. That is the co-registration, is the registration of the EG signal during application of TMS pulses. They are phase-locked neural activity generated for a few hundreds of milliseconds after the TMS. It is well established from early TMS-EG studies and from TMS fMRI studies that TPs include several components and peaks that are generated by the activation of remote areas connected to the targeted region. And the overall TP response is predicted by the structural connectivity of the network in which the target area is embedded. Therefore, as a functional measure of cortical activity, TPs can be used to understand effective connectivity within networks. But TPs have also a key advantage in the high resolution of the EEG. Let me explain you why. If the activation is induced in the target area by the electromagnetic field and then spreads to connected areas, by looking at the TEP's components over time, it is possible to follow the activation of the network as it unfolds. And if we consider the early components, we should be able to detect the responses in those areas that are directly connected to the target. And therefore, we should be able to extract information about specific connections. Specifically, by looking at the latency and the amplitude of the TP components, we could infer conduction delay along the tract and strength of the connection, just as we do with the latency and amplitude of motor-evoked potentials to measure conduction delay and strength of connection in the corticospinal tract. So we should be able to do the same for cortico-cortical connections. In our first attempt to apply this approach and develop this approach, we have focused on the corpus callosum for two main reasons. First, the corpus callosum is the major tract connecting the hemispheres, and despite it has been studied for decades now, we still don't know its function. Second, neuroimaging studies have shown that the structural impairment of the corpus callosum in several neuropsychiatric disorders, like schizophrenia, major depression, and Alzheimer's disease, but we don't know how these alterations are related to the functional impairment in patients. Moreover, we focus on motor areas. That's because it's easier to stimulate these areas and to set TMS parameters compared to other areas. And I will show you later, TMS parameters are very important to get the connectivity measures. So here's our question. Can early TPs reflect effective connectivity through the corpus callosum? In our first study, we collected DTI measures and TPs and ipsilateral silent period in an ipsilateral silent period paradigm. This is because the ipsilateral silent period is an established measure of interhemispheric inhibition that is collected from the hand ipsilateral to the stimulation. So our hypotheses here were to find an association both between the microstructural integrity of the corpus callosum and the TP, and an association between the TP and the ESP, as indicated by the blue arrow and the red arrow, respectively. And we look at various studies to estimate when we should expect a transcallosal activation in the contralateral motor area. In the motor system, the transcallosal conduction delay has been estimated with different techniques that you can see in the slide. And they've shown slightly different results, but overall, the transcallosal conduction delay is in a range between 7 and 20 milliseconds. So at this stage, in the TP response, there is a component that is called N15, and is located under the coil. But we notice that at the same latency, there is also a positivity that is on the contralateral hemisphere. This potential has been mostly neglected in current literature, possibly because it has been interpreted as the other side of the dipole that generates the N15. But we tested an alternative hypothesis that this positivity could represent a different activation. So we call this positivity M1P15. So the results of the first study supported our hypothesis. As you can see here, the latency of the M1P15, okay, here, showed an association with the mean diffusivity of the body of the corpus callosum, which is that part of the corpus callosum that connects the motor areas. And this relationship was not present for other parts of the corpus callosum, like the genu and the splenium. Also, the relationship between the amplitude of the M1P15 and the ESP area was significant. So the higher the amplitude of the M1P15, the stronger the inhibition in the ipsilateral hand. This effect was also replicated in 2 more studies with similar ESP paradigms. One was a pre-registration and one was a register report. Therefore, the relationship between the P15 and the ESP has been replicated several times and is a robust effect. Oops. Okay. Importantly, we did not find these effects for the N15, suggesting that the M1P15 represents a different process than the N15. Another evidence that the M1P15 represents a contralateral response to the TMS comes from a previous studies of my group in which we showed that the interhemispheric functional connectivity measured in the alpha band with the weighted phase lag index in the pre-stimulus window, so before the TMS, was correlated with the amplitude of a contralateral positive component occurring at 25 milliseconds. Now, someone of you might think, well, this occurs at 25, while I stressed that the latency was 15 milliseconds, so could not be the same component. But let me show you how the TMS parameters are important for these measures. And please remember that in this case, we did not use the typical current direction of APPA for biphasic stimulation, but we were using the reversed current direction of PA-AP current direction. So in this study, we ran a study to test if the M1P15 is affected by TMS parameters as current direction and pulse waveform. So we use both biphasic and monophasic stimulators. The current direction determines which neural population is most activated by the pulse in the target area, and indeed affect other measures like motor threshold and MEP latency. And this effect is more strong for monophasic stimulation. So here we tested if these parameters are important also if we want to obtain connectivity measures. In other words, we explore if the pulse waveform and current direction determine which connections with other cortical regions are preferentially activated by the TMS. So we calculated the microstates to test how topographies of TEPs change over time across conditions, as topographies reflect the activation of cortical sources. So as you can see in the slides, the sequence of microstates for biphasic stimulation is very similar across conditions, so doesn't matter which current direction we are using. But if we are looking at the monophasic stimulation, the sequence of topographies in the early part of the response changes quite across conditions, indicating different patterns of cortical activations. When we looked more specifically at the M1P15, we found that it was generated in all conditions of biphasic stimulation. Sorry, here, I'm getting a little bit confused with the mouse. Okay, so it's there for all biphasic stimulations with no changes in amplitude, but with a change in latency, so that the latency of the P15 is longer, and it occurs around 20 milliseconds if we use the PA-IEP current direction. In the monophasic stimulation, the M1P15 was identifiable only in the AP current direction, with higher amplitude and again longer latency. Interestingly, we did not find this component for LM and PA current direction. And previous studies have shown that for these current direction conditions, we need a higher intensity to elicit the ipsilateral silent period. Again, supporting the relationship between P15 and ESP. So this is important. It shows how it is important to control for the simulation parameters if we want to obtain this measure. Finally, we tested the link between the M1P15 and motor behavior. So in our experiments, we use different motor tasks. Across our studies, we have compared several conditions. We had a comparison of isometric contraction in which participants were either relaxed or contracted with their contralateral hand. We also compare a condition of unimanual task in which participants either were resting with their contralateral hand or they were performing a unimanual finger tapping. And finally, we compared a bimanual task in which participants perform either repetitive finger tapping or more complex sequences of finger tapping. And across all these conditions, the only comparison when we found an effect on the P15 is for bimanual tasks. So here, as you can see, the P15 is higher in amplitude for the sequence condition than the repetitive tapping. And interestingly, we found a similar effect over the ipsilateral silent period, in which the ipsilateral silent period is higher for sequence if recorded during sequences than finger tapping movements. So overall, we have shown evidence the M1P15 is a response of the transcallosal connection of M1s. We have evidence from the ESP, ipsilateral silent period, the mean diffusivity of the corpus callosum, the pre-TMS alpha-band interhemispheric connectivity, and the modulation of bimanual tasks. These studies, now we consider these studies a proof of concept that early TEPs can measure remote responses of directly connected areas and support the approach to measure TEP features as latency and amplitude to study effective connectivity of specific tracts. The implementation of this approach will allow to study direct effective connectivity also from other areas and possibly also from other tracts. So to end the presentation, I want to thank you all for your attention, and also want to thank my collaborators that are listed in this slide, and mention specifically Agnese Zazio, who has been carried on a lot of this work, and also Marco Bove and Laura Bonzano from University of Genova for their work on the first study. Thank you all for your attention.
Thank you, Marta. Are there any questions from the audience? Okay, I would like to ask you something. We know that EEG is a very complex method to measure single sources response. So the problem is that it's very difficult to disentangle whether one source is active separately from another. I would like to ask you whether you have ever thought, or are you doing any experiment where you can specifically disentangle with a condition the activity of P15 and N15. So is there any, I was thinking maybe, for example, having a pair pulse TMS, whether you stimulate the source of P15 to see whether it responds differently if you have a pre-stimulation on the M1 of the contralateral side, for example, or other things.
Yeah. Thank you for the question. This is a very important point, because I think the point where we are a bit struggling is actually to disentangle N15 and P15. And we have been thinking about per pulse protocols, or also tasks where we expect an interhemispheric modulation, but not an ipsilateral modulation. I think the tricky point is that both these components, or these peaks, are not the direct activation of the targeted M1. And so they could easily be modulated in the same way by several conditions, even if they are not coming from the same source. So it's quite tricky to find the right condition to disentangle them, yeah.
Yeah, it's a bit a problem for almost every EEG study where you have areas that are co-activated in some way. Are there any other questions? No? I know that you wanted to ask.
Yeah. Maybe I can ask you about the pre-stimulation functional connectivity and TEP latencies. So I was wondering whether the only effect that you find is for this 25 milliseconds component. Did you look for the others?
No, we also found effects in other components. Here we found that we actually look only at the components within 80 milliseconds. We found effects for the N15, the P25, and the P16.
Okay, and
so there are more components that are.
And for instance, what is your interpretation for the P60?
Well, there are let's say that the current interpretation is that the P60 is somehow related to the reafference from the motor twitch. But although this is a quite widespread interpretation, the data to support this interpretation are not very strong yet. I mean, there are studies who have shown a relationship between the P60 and MEPs, but they've not been replicated, and I think in this field the replication of results is crucial, otherwise we don't actually know if the effect is robust.
Thank you.
Since I can't stop thinking about disentangling these sources, have you thought about checking the pre-state of the two sources? How is the pre-state? And then divide in a kind of informed, EEG-informed TMS stimulation, where you have the two sources that are in the same state or in different states, and then to see whether they would...
Yeah, this would be something that we would like to do. It's a bit challenging because I guess you have to, well, first you have to have like a closed loop system, and we don't have it at the moment. But also we will need a lot of trials as well, and we need to control the connectivity between the areas and also the power of the local power of the two areas. But this would be very interesting, yeah.
Thank you. If there are no other questions, we can move on. Thank you very much, Marta.